and we're going to get started. We hope that you'll find today's presentation, Genetic Testing, Genome Sequencing A to Z for Mitochondrial Disease, useful as you're navigating your journey with mito. And we hope today's presentation will answer a lot of your questions about diagnosing this rare disease. Today's presentation will be recorded and available on the Mito Action website under our Mito Monthly Expert Series page, including the slides and the audio recording. I encourage you to take a look at the various recordings while you're there, as I'm sure you'll also find additional topics that may be of interest to you. As we go through today's presentation, if you have questions, feel free to submit them using the Q&A feature. You can access that feature on the top right corner of your screen or through the menu bar. If you're calling in via phone, you can email your questions to info@mitoaction.org, and we'll have the Q&A at the end of today's presentation. So we're excited to have with us today Christine Stanley from Variant YX. Christine is the Chief Director of Clinical Genomics at Variant YX, a genetic testing lab that specializes in analysis and interpretation of whole genome sequencing data. She is particularly familiar with the complexities of interpretation of genetic data for patients with mitochondrial disease, given the many cases she has worked on over the years, first at Athena Diagnostics and later at Cortigen Life Sciences. Christine brings that experience to Variant YX, where she now oversees clinical genomic interpretations for the laboratory, developing the standards for identifying, reporting relevant causal variants in both the nuclear and mitochondrial genomes. In addition to having published articles on the genetic causes of mitochondrial disease, Christine is a member of the expert review committee for the Mitochondrial Disease Sequence Data Resource Consortium, as well as a member of the Mitochondrial Disease Variant and Gene Curation expert panels for ClinGen. She is an enthusiastic advocate for utilizing the best available genetic testing to help end the diagnostic odyssey for patients and their families living with rare diseases, including mito. Christine holds a PhD in human genetics from the Medical College of Virginia with fellowship training at Boston University in clinical and molecular genetics. She is a diplomat of the American Board of Medical Genetics and Genomics and a fellow of the American College of Medical Genetics and Genomics. She additionally maintains a part-time appointment as clinical laboratory director of Q&A Diagnostics. Please join me in welcoming Christine.
Thank you so much, Kira. Thank you for inviting me. Hopefully I can bring some insight to genome sequencing through my experience at both Variantix and Q&A Diagnostics, and just discuss how genome sequencing, which is a type of genetic test, is utilized in the clinical laboratory and how it applies to diagnosing mitochondrial disease. So I'm going to take time here to talk about, have a little bit of an intro and closing, but the main parts of the presentation are the DNA, talking about DNA sequencing for mitochondrial disease, giving you a description of what that is, just DNA sequencing in general, and then how genomic sequencing is utilized in diagnosing patients with mitochondrial disease, and then provide some clinical case examples that will hopefully highlight that part of the discussion. So if we go into the background of mitochondrial disease, just basically mitochondria are always described as being powerhouses or energy factories of our cells and organs. And so what happens to the mitochondria happens to our bodies, right? So if the mitochondria aren't functioning well, it makes it very difficult for our bodies to function well. The organs that require large amounts of energy are usually the first affected and the most severely affected, like the brain, the muscles, the heart, and the lungs. But other organ systems can also be affected, like the kidneys and eyes, liver, and pancreas, etc. And typically when you see more than 3 organ systems involved in a disorder, you start to think that this might be a mitochondrial disorder. So like I said, if more than one of these energy demanding organs are affected, they can result in some of these clinical symptoms that might have your clinician suspecting that this is mitochondrial disease. Developmental delay with some other organ involvement, cardiomyopathy, high levels of lactic acid in the system, ophthalmoplegia or ptosis, such as eye findings, hearing loss, and severe gastrointestinal dysmotility, or developmental regression in the presence of other illnesses. Some of these disorders that are associated with mitochondrial disease are also associated with different types of disease. So it is very tricky to try to determine whether somebody has something that seems like a presentation that's arising from the mitochondria not functioning properly to other disorders that can mimic mitochondrial disease, or if you look at the other way, mitochondrial disease can mimic other disorders. So you'll see that failure to thrive, short stature, developmental delay, and muscle tone can be associated with mitochondrial disease, but it can also be associated with other genetic disorders. And on the basic level, we talk about the inheritance of mitochondrial disease. So there are two ways that mitochondrial disease is inherited. One is from changes in the mitochondrial genome, which comes from the mother, because we inherit our mitochondria from our mothers, since the majority or almost nearly all mitochondria that comes in the egg and not in the sperm. So that's why we get that from our mother. And then there can be changes in the nuclear genome that also that's the standard genetic testing that you would have when you think about genomic testing is coming from the nucleus of the cell, and that's the 46 chromosomes, where we got 23 from our mothers and 23 from our fathers. So those nuclear genome changes do occur where they are in genes that are utilized by the mitochondria. They're either transported into the mitochondria or they have some relationship with other genes within the mitochondria. So if you have a pathogenic change or a change there that's causing disease, it can also present as a mitochondrial disease, even though that change didn't happen within the mitochondrial DNA. So there's two types of sequencing, or there's multiple types of sequencing we could talk about, but lately the diagnostic labs have moved from the standard, or the used to be gold standard of Sanger sequencing, which was very limited in how many genes you could actually sequence, and it was a tremendous amount of work to sequence more than one gene, to something called next generation sequencing. And then next generation sequencing is just an ability to sequence a lot more with a lot less resources. You're sequencing things hundreds of times or even thousands of times rather than twice when you do a Sanger sequence. And this sequencing has been applied to mitochondrial disease, and we'll just talk a little bit about what is sequencing. So basically, we're talking about what's in an individual's DNA, whether that DNA comes from the nucleus of the cell or whether that DNA comes from inside the mitochondria, which are organelles within the cell. We're doing the same thing. We're reading what the sequence says, the spelling. So there are 4 bases in DNA, and the shorthand nomenclature for them are A, T, G, and C. So we're really looking at the letters A, T, G, and C, and the combinations of those letters spell out the recipe for how the cell is going to put together proteins, and directs basically the development of the cell in the body. So when there's a change in one of those letters, it can change the what's read, and it can change what kind of protein is made and whether that protein is functioning properly or not. There's a lot of changes. We all have a lot of changes in our DNA. Every so often there's a change that occurs in a very, very critical region, and that will cause disease in some individuals. But we all have new changes that arise in us. We have 1 to 2% of our genome is actually coding for proteins. And so, 98, 99% of all of our genetic material lies outside of those regions. So if we have changes there, they're less likely to impact us than if they happen in these very important regions. So even though we all accumulate these changes, it's kind of part of evolution. When it's just a kind of matter of luck whether they hit in critical regions or not. So when we are extracting the DNA out of the cells to try to read it, we're basically just processing the DNA through some machines with a little bit of manipulation, basically that reads out a sequence of letters to us. And then we use clinical software tools to look at that sequence of letters. So the DNA, like I said, is located in 2 places within the cell. They're both in the mitochondria and the nucleus of the cell, and they're both important for diagnosing mitochondrial disease. In the mitochondria, they can have multiple copies of each mitochondrion can have multiple copies of its small circular genome, and then you have multiple mitochondria in the cell. So you're pooling together DNA that's from multiple mitochondria that's in the cell. And then for the nuclear genome, you're pulling the DNA out of the nucleus of the cell. And you can either do those separately, where you're just doing a mitochondrial DNA test, or you can do just a nuclear DNA test, or you can pull that DNA out together, and you can do sequencing that will evaluate the genetic code in both the nucleus and the mitochondria. The mitochondrial DNA contains a very small number of genes relative to the nuclear DNA. The mitochondrial DNA contains 37 genes, or codes for 37 genes. And a gene is just that segment that codes for a protein, or other types of functional gene products are like TRNAs that are used to transfer the building blocks of a protein to create a protein. And there's something called ribosomal RNA that's also important functionally. So you'll have protein. Usually you hear of genes and proteins together, but genes can code for other things besides proteins. And basically these are products that we need for our body to function well. In the nuclear genome, there's about 20,000 genes total, with over 1,000 of those genes encoding some element that's important for mitochondrial function. So like I said, the genome sequencing is a type of a genetic test. And so there are many benefits to performing genetic testing from a diagnostic purpose. Number one, it provides an answer. So we're trying to understand what maybe, somebody has a child or family member who, or they themselves are experiencing clinical symptoms and they don't understand why this is happening. Just getting that genetic diagnosis provides an answer to that. It ends the further testing. So once you have your diagnosis or your answer, you don't need to continue to be tested. You've got the answer. You can now focus on that particular disease and maybe reach out for other connections to be able to move forward with your clinical management. Sometimes knowing the actual change and what gene it is in can guide treatment. And it can also inform you on agents to avoid. So for example, patients who have changes in the POLG gene that is associated with mitochondrial disease can't take valproic acid because it will adversely affect their liver. So even if there's no cure or treatment for a genetic disorder, knowledge of what the genetic change is and what gene it is can help us to avoid treating with things that would exacerbate the problem. It empowers, a genetic diagnosis would empower connections both at the family level, so it allows you to participate in support groups, family support groups with individuals who have similar or the same genetic disorder, and also can empower connections beyond support groups to maybe researchers who are researching that particular gene, or if you're fortunate enough to see that there's a clinical trial for a particular disease that's associated with the gene, it allows you to make those connections. So basically it's providing many options to the patient, the family, and the clinician for the patient's clinical management. So that's just genetic testing in general. For genomic testing, there are added benefits of doing a whole genome test, which I'm going to talk to you today about. Genomic testing is just more comprehensive. It detects more variant types. So when I talked about that sequence, those letters that we're reading, that's one type of change. If one of those letters was an A and now it's a T, that's called a sequence variant. It changes from what we'd expected it to do to something else. Most sequence variants are not causing disease, but there are a percentage of them that do cause disease. There's other type of variant that's called a structural variant. So if you like think of the whole chromosome, rather than one tiny letter of that DNA being changed, some big chunks of the chromosome are missing or duplicated or moved to a new location, that type of thing. And those are called structural rearrangements. And those are variants that you can detect using genome sequencing. There's also repetitive sequence. So when you're talking about the ATGC, you have, they're sort of randomly, it's not random, but you know, you can say AAGC, and then there are regions though that are very repetitive. So it'll be CAG, 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 and it goes on. And in an individual, like in the general population, they might have a range of those CAG sequences of like, say, for example, 20. But then we'll look at an individual who has presenting with clinical symptoms, and we'll see they have 200. And so it'll be defined in the literature for whatever that particular gene is, that if you have under, say, 50, it's a normal population variant. If you have more than 50, then you have a genetic disorder that's a result of a repeat expansion. And that is a type of variant that you can detect using genomic sequencing only if you have developed a software tool that's very good at detecting that type of change. As we talked about, you can detect mitochondrial variants, which is very similar to those sequence variant changes, but this is happening in the mitochondria rather than in the nuclear genome. And then there are variants that, like I said, a very small percentage of our whole genome, like 1% or 1.5% of it is actually coding for genes. The rest of it is, you know, the stuff that's in between the genes, or a gene is coded for by like different pieces of sequence that are called exons, because they're the sequence that's expressed. And so when you, you know, there's some space in between one exon 1 and exon 2, and that's called intronic sequence. It's like the sequence that intervenes. So our molecular biology is very interesting in that it doesn't just code for one straight gene. Actually, in the mitochondria it does, but in the nuclear genome, it takes the gene and it like breaks it up into pieces, and then it puts spacer DNA in between, and it makes the body have to put the puzzle pieces together again. So there's changes that can occur in that spacer stuff that makes it challenging to put those puzzle pieces together again. So those are called, that's the intron, and those are called intronic variants in there. We don't know a ton about them right now because we don't typically sequence that region. So there's not a lot of information about it, but as more and more studies are utilizing genomic sequencing to perform their analysis, they're starting to find more things that are out in the introns that are actually affecting how that protein is put together. The other benefit of genomic testing is there's lots of types of genetic testing. There's panel, gene panel, single gene. You can put a bunch of genes together and call it a gene panel. You can do all of the expressed sequence or the full exome. And that's all 20,000 genes, or you can do genomic sequencing. So if you do genomic sequencing, you've done everything. You've got all of the genes and everything in between. And that allows you to be able to reevaluate using like a digital way rather than having to get another blood draw and submit another sample and wait for the lab to do their sequencing. So that's one advantage. That gives you a single turnaround time. So sometimes what happens is if your clinician orders a single gene test or a small panel of genes, they'll have to have like, you know, you have to process that sample, and then there's whatever, like a 6-week turnaround time, and then you get the results and you have to go back in and see the clinician. They have to explain, oh, this was negative, so now we're going to do this other test, and then you get another blood draw, and et cetera, et cetera, et cetera. So you end up with these long cumulative turnaround times that extends the time to the diagnosis rather than doing it all at once. And if, say, like you do a gene panel and the next week a new gene has been described, it wasn't sequenced on that panel maybe, and so you would have to then do it again to get that one more gene. So this avoids that one more gene thing, because the industry is moving so rapidly with understanding connections of genes to disease that you know, it makes it very challenging to keep up with it from a lab perspective, where you'd have to keep building these gene panel tests. The other thing is the data can be reused for other types of testing. It's not something like, you know, something down the road, maybe you want to know whether you have any hereditary variants that put you at risk for hereditary cancer, or if you're at a point where you want to have children and you want to know if you have a reproductive risk of carrying a recessive pathogenic variant, you could do carrier testing, or there's pharmacogenomic studies, and also you could potentially utilize the data for other purposes. We don't do pharmacogenomics yet at Variantix, but it's there if in your, you know, you could potentially use it for other things. So when we're talking about using the genome sequencing for mitochondrial disease, I kind of touched on that the typical path right now, and a lot of this is driven by insurance companies. They want to see that you tried some smaller, cheaper tests first, and those were negative, and then you would move your way, you know, up to from like a small single gene test or a small panel of genes to exomes, and then if that's negative, maybe to genomes. But you know, it gets more and more challenging to get them to cover that if you go in that you go that path. But it's been traditional because it's been really dictated by the insurance industry. We can flip that upside down and say, let's start broad. Let's start with the most comprehensive testing and then like reflex down. Now, I would never reflex down technically from a genome to an exome, but you might, if you don't have like, there's some reason why the genomic test doesn't cover a particular region that's very likely to be associated with your clinical symptoms, you could potentially go down to a single or smaller test. And genomic sequencing is very comprehensive, but it's not 100%. So there would be other tests that could fill in. And then you also might want to do other tests that might confirm it. So if you found something using the genomic sequencing, like we saw this change here, there's not a lot in the literature about it, we're not sure. There may be some kind of confirmatory smaller test that you could do, like if this was a gene that would be predicted to result in absent enzyme activity, and there happens to be an enzyme activity test for that gene, you could show that, oh, see, we have this change, and then we ordered this other test and it has 0 activity. So it's very likely that that change is what's causing the clinical disorder. When we talk about sequencing, either if you're talking about doing gene panels, exomes, or genomes in the whole next generation sequencing type process, they all start with the same starting material. So you'll have a blood sample or a salival sample, but you'll extract the DNA from that sample, and then the DNA gets chopped up into pieces. So all of these tests will start with the same thing, fragmented genomic DNA. The difference between panels and exomes and genomes is that because the genes are a small percentage of the total DNA, you have to fish out the important pieces, right? So you're fishing out all of the 20,000 genes if you're doing an exome. And like I said before, that each gene is broken up in your DNA into like pieces of a puzzle. So you're actually fishing out parts of each gene. And for genomes, that's not a step that you do in the laboratory. So when you do sequencing for genomes, you avoid this one step. And the reason that's important is because whenever you have to manipulate the DNA by like pulling pieces of it out, you just like fishing, you're never going to catch like exactly the same number of every kind of fish, right? You're going to catch more of some and, less of another. You might even miss some altogether. So you end up biasing your like data set because you've had to like fish out the important pieces, and not all of them are as easy to capture. And I talked about the different types of variants that you can get from whole genome sequencing that you can't really get from exome sequencing. And this is just a list of some of those variants. So we talked about the single nucleotide variants that you can get for both nuclear and mitochondrial genes. You can get that from, exomes and genomes. But the challenge for exomes is something called short tandem repeats, and I said that CAG, 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 that repetitive sequence. To be able to count those, it's very difficult to do with just exome sequencing. And then structural variants are also very challenging to read off of exomes and panels. Both of those would be really difficult to get all the structural variants. So their structural changes where there's missing pieces or extra pieces come in a lot of different sizes. They're a very, very important type of variant to be able to identify, because a lot of times when you have a big piece missing, you're missing a big part of the gene. So it's like more likely to be associated with disease than some of these other types of changes. And so, the ones in the black seem to be pretty well detected using either genome or exome, but the variants listed in the red are really either much more efficiently called in genomes or can only really be called by looking at genomes. So these other regions, these small changes where you got extra material or you're missing material, are typical blind spots for exome sequencing. So to highlight some of the benefits to doing genomic sequencing over exome sequencing or panel sequencing, we've got put together a few clinical cases to review so that kind of see how this is applied. So in the first case is of a 51-year-old woman who has some muscle weakness and myalgia, has difficult speaking, and is choking. She's in her when she was in her 40s, she started to develop ptosis, which is like a droopy right eye, and she had surgery for that. And then at 49, she started to develop some skin rash. Clinical neurological exam was normal. Her blood lactate levels were normal, but her muscle histology showed ragged red fibers, which could be indicative of a mitochondrial disease. She had genomic testing, and there was a mitochondrial variant. So that would be a change in that little circular DNA that was in the mitochondria in something called a tRNA. And tRNA is a transfer RNA. It's this product that is a product of a gene that's not a protein. It's a product of a gene that's an RNA, and the RNA helps to build proteins. It's kind of one of the worker bees in the process of putting together proteins, but it's not a protein in itself. And so this change was what was causing disease in this case. And then similarly, there was another case of a 25-year-old woman who had muscle fatigue, headaches, had recurrent episodes of stroke-like episodes, and kind of a paralysis on the right side of the body. There was some abnormal lab results and abnormal muscle biopsies. And she also had this autoimmune disorder that looked like Hashimoto's thyroiditis and Addison's disease. And this case was another change in the mitochondrial genome, which was a variant in the ND4 gene. And one thing I didn't talk about yet, which is a little bit more challenging for mitochondrial changes than nuclear changes, is that because you have a bunch of different mitochondria in a cell, they can have differences in the DNA from one another. So you will have these populations of different types of DNA. The nuclear DNA should be basically the same when you pull the DNA out of all the nuclei of the cell. But because you're pulling it out of different mitochondria, you can have differences, and you'll hear this referred to as heteroplasmy. So if all of the mitochondria have the same change, and so you see it in every copy of mitochondrial DNA that you see, then they'll call that homoplasmic. And then if you have differences, 90% have what we would consider a normal sequence, quote unquote, and then 10% have an abnormal sequence, we would say that for that abnormal variant, it would be 10% heteroplasmy. In some mitochondrial diseases, you have to have homoplasmy or near homoplasmy in order to be presenting clinically with the disease. And in some disorders, it depends on the level of heteroplasmy that you have. And then another complicating factor with mitochondrial disease is because you have that distribution of mitochondria, if you could just picture the egg has a bunch of hundreds of thousands of mitochondria, and then when the egg fuses with the sperm, then you start to develop the embryo develops, and those mitochondria can get distributed differently if there's a change. Those mitochondria that have the change might go into some cells, but not into others. And eventually this all develops into different organ systems. So you can have a high level of heteroplasmy because you just randomly it distributed that pathogenic change, that's causing disease into the mitochondria that all went into like the development of the muscle, or all went into the development of like the ectoderm, which involves the neurological tissue. So you might test someone's blood and you might see very low level evidence of a change there. But then if you test the skin or some other tissue type, you'll see that it's a different percentage of that change. And that's because you have so many mitochondria within your cell, and they can randomly distribute to different organ systems. And then after development, they're all replicating. So you'll get the continued presence of that change in those mitochondria that are in that particular tissue type. So these are both the classic mitochondrial disease that are caused by a variant in the mitochondrial genome that's causing a problem with one of those 37 genes that are encoded by the mitochondrial genomic DNA. Another case here is a 5-year-old girl with bilateral club foot, mild facial dysmorphism, some mouth and teeth abnormalities, developmental delay, so the weakness or hypotonia, had difficulty speaking and spoke very slowly, and then had they considered mitochondrial disease because she had a constellation of a failure to thrive, the weakness or hypotonia, dysarthria, and a tremor. She also had abnormal biochemical results with the elevated plasma lactate, lactic metabolic acidosis, elevated alanine levels, and elevated urine organic acids. So these are kind of results that would have you suspect mitochondrial disease. In this case, though, the variant was in the nuclear genome. So this is a heterozygous or 50% change on one type of variant and 50% on the other, which is like typical for recessive disorder. So it was in the FARS2 gene, which codes for an enzyme that regulates a phenylalanine transfer, so that tRNA that transfers over amino acids to build proteins. So one variant was inherited from the father, and one variant, which was a deletion type of variant, that big bits missing, was inherited from the mother. So you need to, if you have one copy of this gene, you're fine. So both the mom and dad were fine, because even though they had one change that was would have particularly caused disease, they had another copy, because we have 2 copies of almost all of our nuclear genes, except for those on the X and Y chromosome. That was enough for them to be functionally normal, but they each passed down the pathogenic variant to the child, and then the child was affected. And this is a classic case of a recessive disease, but it's autosomal recessive, so it's coming from the nuclear genome, but it's presenting as a mitochondrial disease because of its important role in the mitochondria. So the take home here is these variants would not have been identified if you just looked at the mitochondrial genome, and that in this particular case too, there were 2 different types of variants that are also pretty challenging to identify on, like see even an exome. So there was one that was a single, letter change, and then there was one with the larger piece of information missing. So those are not something that you may not pick that up on your traditional testing. But if you do whole genome sequencing, you can pick those up on a single test and get a single result report. Case 4 is that of a 24-year-old woman presenting with the clinical symptoms of meat loss from age 15 on. So she had stroke-like episodes, seizures, and high lactic acid levels. Genetic testing identified a pathogenic variant in the mitochondrial genome. Or detected a variant in the mitochondrial genome, but the mother, because you inherit your mitochondrial DNA from your mother, the mother was tested, and she actually had higher heteroplasmy of that variant. So, and she didn't have any symptoms. So there was a question as to whether this was really the sole cause of the clinical presentation. The woman had an ataxia, headaches and tinnitus, and said weakness as well, and she was having having a tremor that worsened with movement. Some additional clinical symptoms: loss of peripheral vision, speech articulation issues, renal failure, and contractures. So what was identified here? So we had the mitochondrial variant that we had seen, but then there was another variant in the nuclear genome, which was on a gene called VARS2, which codes for an enzyme that regulates tRNA. So the reduced function of the VARS2 due to the nuclear variant and then the mitochondrial variant seemed to be the explanation for the patient's symptoms. So analyzing just one, either the nuclear or the mitochondrial, would be not sufficient to give you all the information that you would need to explain the patient's clinical presentation. And so you really need a test that connects both the nuclear information and the mitochondrial information. And that would be whole genome sequencing, as long as the whole genome sequencing was sequencing the mitochondrial genome as well. So just to conclude, mitochondrial disease is really challenging to diagnose because you have so many different organ systems affected, and the changes can be caused by the mitochondrial DNA or the nuclear DNA. And also like the heteroplasmy levels can also complicate the diagnosis part. Genetic testing is a recommended first step in diagnosis for mitochondrial disease in general, but when you're doing genetic testing, you want to pick the most comprehensive genomic test that you can do so that you would have that shorter path to diagnosis rather than having to reflex up. Unfortunately, it's not always possible because insurance companies may require you to do those smaller tests first before you go to genomic testing, but we're really expecting that that's going to all flip, where we're going to do the broadest, most comprehensive test first so that we can really shorten the time to diagnosis and actually shorten the out-of-pocket costs as well. And genomic testing can identify causes that aren't in the mitochondria but have overlapping clinical symptoms. So I didn't really talk about this, but at the beginning we talked about some of the clinical symptoms that could be mitochondrial disease, but it could also be something that's not mitochondria. So if you're really just focused in on a mitochondrial panel and you have a disorder that presents somewhat like a mitochondrial disease, but it's not actually one of the genes that are involved in the mitochondria, it just has clinical symptoms that are similar, you would miss it because you wouldn't be looking for that type of gene. So if you do genomic sequencing, you're getting all the genes that are in, all the other diseases, including all the mitochondrial diseases. And there's another thing too, there was a study, an Exome study that saw that like 5% of the population with, the severe clinical presentation actually had 2 genetic disorders going on at the same time. So when that happens, the clinical symptoms, it doesn't really point to a single disease because you've got this combination of 2 disorders together. So if you do genomic testing, you'll be able to identify patients that have that, unusual presentation where they have more than one genetic disorder happening at the same time. To order genetic testing, it's genomic testing. It's still, there's no direct to consumer. So your physician has to be the one who orders the genetic testing, and they'd review a consent form. So you would both sign it, the test requisition, and they'd explain the limitations of the testing, etc. Your insurance company may or may not cover genetic testing, so you would need a pre-authorization to see if it would be covered. The benefit to genomic testing is that if you have a negative or inconclusive result, you can request a repeat analysis of the data that doesn't require another blood draw. It just requires the laboratory to re-look at the data in light of new information that would be out in databases. So as everyone's doing this type of testing, they're rapidly depositing information into databases. And then that's the data that we use to interpret a new patient's results. So if you had a negative result, but tomorrow the cause of your disease was published, then, within a few months all the databases would be updated, and then our clinical software tool would be updated. So if we reprocessed that result, we would then see the connection. So the benefit of the genomic sequence information is that you can keep reanalyzing that information. You're allowed to receive copies of results from your ordering clinician, but legally you can also have the right to receive your test results directly back from the diagnostic lab if you request it. It's something that people just don't necessarily know, but it's a possibility for you to do that directly. If you order testing from a laboratory, ask them if they can detect all variant types, including deletions, duplications, and short tandem repeats, if they sequence both the mitochondrial and nuclear genomes. So you want to try to get the most comprehensive test the first time around so that you have a better chance of getting the answers. So those are some important questions to ask the laboratory. So that was it. If you're interested in learning more about genome sequencing at Variantix, we have some information on our website at the link there, or you can send an email to us at variantix.com. One of the things that that I didn't mention was that in order to do testing and to validate testing and to make sure that you have a really good test, you have to have a lot of controls to do that. And one of the things we're looking for us is we can do this, the mitochondrial sequencing, and we had a lot of controls to show that we're very good at detecting any variant changes there. We had a limited number of patients or samples that had big deletions or big missing pieces from the mitochondrial genome. So if you or you know anyone who's been diagnosed with the large mitochondrial deletion, we're recruiting patients so that we can perform whole genome sequencing and show that our test can pick those up as well. And then you would have a genomic test result report as a as a consequence of that. So please contact us if you have or know of anybody who has that result that would be interested in whole genome sequencing.
Thank you, Christine. I appreciate it. That was really informative. And as you know, I mean, the diagnostic journey is one of the most difficult parts of this journey. And statistics show that our patients, see between 8 and 10 doctors and an equal amount of time, 8 to 10 years before a lot of our patients receive diagnosis. So to know that the genome sequencing is more comprehensive, and if we can start there, hopefully that will help to reduce the time to diagnosis. So this was really, really informative. So we've got a few questions that came in that we'll ask, and just want to remind everybody, you can submit your questions through the Q&A panel on your screen, or you can email them to us at info@mitoaction.org if you're listening on the phone. So the first question is saliva as definitive as blood when you're doing genome sequencing?
It's a really good question. We validated to be able to do saliva or blood. Is it as definitive? Yes, pretty much. I will just say from the lab analysis side, saliva can produce more artifacts in the sequence. So sometimes we'll see something that will look a little suspicious, but when we look at the data, we'll see that's probably coming from some extraneous material that was in the saliva that we happen to sequence. So it should, when you're doing a thorough analysis and you're not tricked by any kind of like artifacts in there, they're just as comprehensive. But my preference is to look at data from whole blood because it's cleaner and it makes an easier time for me to do the analysis.
And along those same lines, can the tests with the saliva be repeated without having a second sample?
Yeah, if you have a successful genomic sequence of saliva, then, if you're like the lab offers it as a validated sample type for their testing, then you would have the same issue where you don't need to do a repeat sample. As long as they could get it, as long as the sequencing was successful, then they shouldn't require a repeat sample. You should be able to just do reanalysis of the data at that point.
Okay. Next question is, can you share a little bit about the benefits of having other family members tested?
Oh yeah, that's a really good question too, because we didn't really talk about it exactly, but when we have a variant of uncertain significance, there just hasn't been any data reported in the literature or any databases of a variant. Sometimes for mitochondrial disease, we'll look at the maternal family members and see if there's a correlation between individuals who are presenting with clinical symptoms and the presence of that variant in their mitochondrial genome. And not only that, you can correlate it with percent heteroplasmy. So like if somebody who's very severely affected has very high heteroplasmy level of that change, and someone who's more mildly affected has a lower heteroplasmy level, you can sort of see these correlations. The problem is you need kind of a lot of people to do that. And so it becomes more challenging and costly to be able to do that analyses. But there are definitely like lab groups that are doing that type of testing for changes. And so then if they've done it in a family, then they can then use that information, put it in the database to say that that variant appears to be associated with disease. It just, from a lab testing perspective, we typically don't get enough family members to do a really confident correlation between the variant and the disease. And then the other testing, it's not benefits of like other family members necessarily to make the diagnosis, but that testing other organ types, because in mitochondrial disease, we'd also want to see if like there's a different heteroplasmy level of that variant in different organs. For testing family members for other types of like variants in the nuclear genome, it can be important if there's another affected child to see if they share the same variant, the same thing as that co-segregation of that variant with with individuals within the family that have clinical presentation of disease. It can be useful for trying to interpret whether that change is actually likely to be associated with disease. If we see it in patients that are clinically completely unaffected, then we're less inclined to think that that variant is associated with disease, so we can kind of rule it out. If we see it correlating, then it's the power of how many individuals actually have that variant to see if that statistically makes it more likely to be associated with disease.
Okay, thank you. The next question is, if you've had, excuse me, if you've had exome sequencing done, does it require to have a new sample taken in order to have genomic sequencing done?
Okay, yes. So when I showed the picture of the little kid fishing the DNA, that's the difference between the platforms. So exome sequencing is sequencing only like 1% or a little more than 1% of the total genome. So genomic sequencing sequences all of it. Now, slight disclaimer, it really only sequences 98% of it, but way, way, way more than what you would get in an exome, because there's no fishing out of the pieces of the genes in there. So if you have had exome sequencing, it doesn't translate to genomic sequencing because the sequencing process eliminated most of the DNA. It threw away 98% of the DNA and saved like 1 to 2% of the DNA for analysis. So they already threw it away. You can't use it again. Now, if you already have given the sample, they may have leftover sample that can be used for sequencing the genome, but you can't just reanalyze an exome to get genomic sequence results. But you can reanalyze an exome for new connections. So if you already have an exome, you should be able to do a reanalysis in case there's any new gene connections that potentially weren't there when you first had it performed. So if you do have an exome sequence, most labs have a policy that they'll reanalyze your sequence at least one time within a 1 to 2 year period. So you would go to the laboratory or your clinician and say, you know, when should we reanalyze this to see if there's any new connections? Because the databases fill up so fast with new information. It's such a rapidly growing data sets out there for data analysis, and new variants are going into our databases all the time associated with disease. So something that wasn't there, you know, a month ago can be there. So for reanalysis for exomes, a lot of people wait till like as far as they can until that one, you know, that window is almost up. Like if they give you one reanalysis in a year, then they're going to wait till it's close to a year so they can make sure that they have the most current information. May or may not work. I mean, there's a combination between the data going into the databases and then the data being uploaded into the software tool that's used for the reanalysis. So that takes time. So if you ask for a reanalysis a month later, probably nothing's changed, even if some new stuff came out in the databases, because they haven't updated their software yet. And usually the updates happen quarterly. So genetic counselors tend to wait until close to the deadline of when they can still get the free reanalysis to do it, to make sure they got everything as current as possible.
Okay, great, thank you. So 2 more questions. If you're interested in having a reanalysis, is that something that the testing company does automatically, or is that something that has to be requested? And can the patient request it, or does the physician have to request it?
That's a really good question. So that's going to be dependent on the company. They usually, most companies have a policy, and it would be because it, you know, we're running through CLIA labs, which are the, CLIA is law on how we test patients and we provide answers. A reanalysis, should have to come from the ordering clinician, because they're the ones that the results will be returned to. You have a right to get that result once it's done, but whoever initiates the original test needs to initiate the reanalysis. So basically you just have to bug your clinician who ordered the test to say, I want a reanalysis of this. And then they will reach out to the laboratory and see what their policy is on reanalysis. Now you can call the laboratory yourself and ask for your results. And then you can also ask them what is their policy for reanalyzing the samples and get all that information for your clinician. And then you can say, hey, this lab said that they will do this at no charge if we request it now, and I need you to request it. And then they'll reanalyze it and issue an updated report if they find anything new.
Okay, great. Thank you. And so the last question is, typically how long does it take for the sequencing process? Once you've given a sample, typically how long does it take to run the testing and get results back?
So these are all really very good questions. So the processing is interesting. So it depends on the laboratory and it depends on the type of sequencing you're running. But the equipment to do exome sequencing and genome sequencing and the reagents and the supplies you need to run it are very, very expensive as far as like compared to any other types of lab tests. So what can happen sometimes is if your lab's processing a lot of samples, you can have a shorter turnaround time because you fill up all the lanes on a run and then you can process them. But you might have a lag if you don't have as many samples coming in waiting for enough to run it, because it costs you the same amount to run a full train as the train with just one person on it. So you want to maximize the number of samples on a test. So that's why from the lab processing side of things, there can be a big variability in the length of time it takes to actually do sequencing. It takes about a week, just the wet bench process part of it. If all was good and you could, as soon as that sample came in and it was extracted and run through, you can technically get that done in under a week's time. But because there's sometimes waiting for samples to batch, and then genomics takes a little bit longer to process, and it takes a little bit longer for the data analysis part, but that's just a matter of a few days. And then, then the analysis part. So then everything's all equal when you get to the analysis, and it really depends on the clinical software tools that you have developed or the tools that you are using to do the analysis, because we usually filter the variants that we get through. So we might on a person have like 20,000 different changes in this person, and we have to figure out which of those changes are important and which aren't. So we use like a lot of things like population frequency. If it's a really, really common change, we're going to filter it out. And there's lots of ways to filter and manipulate the data that comes through to try to bring out only the things that are most likely to be pathogenic. And so if you have a really good software tool, it does a really good job pulling out the things that are pathogenic or that are causing disease from the stuff that doesn't cause it. It can be a pretty fast analysis, where the analysis part may only take a week or two. But remember that when we're looking at all these changes, we'll see that they show up in a database, but we have to confirm that that database has accurate data. So once we see that connection, then we go out and we get the publication, we read the publication, there may be multiple publications for that particular variant, then we have to decide what the likelihood of that variant causing disease is. So a lot of the time it takes to do an analysis is really the analysis time. And as we further like sequence more and more people and we get more and more data and we have more definition around what each variant does, the analysis time will shorten over time. But right now we're just getting so much information and we have to go through every variant one by one and look to see what information is available. And then we have to confirm that that's actually good information, because there's some information out there that's not good. So we have to critically review all this stuff. So it takes a fairly sizable, knowledgeable team of people, and geneticists, variant scientists, genetic counselors to really put the puzzle together. So I didn't even mention the part about clinical correlation. So we have genetic counselors who are looking at the clinical symptoms and then looking at the clinical symptoms that are associated with the genes and then seeing if they're a good match to one another. So there's a lot of manual effort that goes on beyond the sequencing. The sequencing itself is really not the thing that usually takes the time. It's usually the analysis part. And if the team is backlogged, they have more cases than they can handle, then you're sort of in queue waiting for that analysis and trying to get that balance of the perfect number of people to analyze the cases, that your caseload that comes in. It's a really tough thing to balance for laboratories. So you might hit like a fast turnaround time one time you send a patient sample in, and then a much longer turnaround time the next time. But all these clinical laboratories, they're all CLIA regulated, and they have to quote what the turnaround time is. So they tend to quote somewhere around 6 or 8 weeks for exome or genome sequencing results, so that they can give themselves enough time in case they have like some of these little changes in process and they get a backlog or something. And if they don't stay within their published turnaround time, they either have to change their published turnaround time, or they have to fix the problem by hiring more people or whatever. So we're really bound as clinical diagnostic laboratories to publish an accurate turnaround time and then stay within them, because when we get inspected, they look at our turnaround times and they see if we're meeting them. So it's somewhere, I think, around 6 or 8-ish weeks for most laboratories. And some offer rapid turnaround times where, you know, that you pay more for the test, but your test goes to the front of the queue and then it's the first one analyzed off. So they can, some labs will offer a 2 to 4 week exome turnaround time, but they charge more for it because they have to like run it no matter whether they have enough samples to batch, and they have to interpret it. That's the first one interpreted, that kind of thing. They tend to offer that for like neonatal issues for a really severe disease.
Thank you. It's incredible what a complex process it is. And like you said, the number of players that are involved in analyzing and interpreting the data results. So thank you so much, Christine. This has been a really helpful presentation. We hope that you know, everyone has learned something today. And if you have any additional questions, feel free, you can send those in to us at MitoAction, excuse me, at info@mitoaction.org. And we can follow up with Christine and get those questions answered for you following the call. So I just want to thank everybody for joining us today. A reminder that the presentation will be recorded, and within 24 hours, it'll be posted on our website under the monthly Mito Expert Series section, along with the slides that Christine shared with us today. I also just wanted to remind the community as well, on January 10th, we will have our annual Mito Town Hall, where all of the members of the Mito community, patient advocacy, clinicians, pharmaceutical companies, all come together to share their highs from 2019 and what to look forward to in 2020. So stay tuned for information on that particular call. That will be on January 10th at 12 o'clock noon, also on Zoom meeting. So we thank everyone again for joining us. Thank you again, Christine, for sharing all of your wonderful expertise in diagnostic. And absolutely. And I wish everyone a wonderful holiday, and we look forward to staying connected with each and every one of you. Have a wonderful day, everybody.
Thank you.